Hello, this is Jim Schnabel, and we are doing the 24th lecture in the series from the book Principles of Biochemistry, authored by Albert Lehninger, 7th edition. Today we'll be discussing all elements of the citric acid cycle. The citric acid cycle has respiration in three different components. In the first segment, the organic molecules are oxidized to acetyl-CoA. In the second broad category, acetyl-CoA is converted to carbon dioxide, and an electron carries NADH and FADH into oxidative phosphorylation. And lastly, the electron-carrying molecules are conserved as energy as ATP. The synonyms for the citric acid cycle also include tricarboxylic acid cycle and the Krebs cycle after Hans Krebs, who in a series of elegant stereoisomeric specific labeling experiments was able to deduce the cycle, which has a background skeletal molecule that rotates through a cycle, and you have addition of pyruvate at one end and then energy and carbon dioxide coming out at the other end. So to begin, we have pyruvate entering the mitochondria where this series occurs via mitochondrial pyruvate carriers. The initial reaction conversion of pyruvate to acetyl-CoA is catalyzed by pyruvate dehydrogenase, which employs 5 coenzymes and 3 separate enzymes. It is a complex. We're going to spend some time with the mechanisms of these reactions. They're certainly more complex than what we had seen in glycolysis or gluconeogenesis. So we'll be spending some time on those. This is the particular beginning of it. There are 3 complexes: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase, E1, E2, and E3. So in the first of these reactions, pyruvate is converted into an intermediate of trihydroxy TPP with elucidation of carbon dioxide. In the second reaction, the oxidized lipolysine, this is a cysteine molecule, that is reduced to free cysteine and an adduct between the sulfur atom and this carbonyl here. So this is a reaction that occurs. You get oxidation of this point right here, and you get an adduct forming. And then in the third reaction, coenzyme A replaces the sulfur right here, and you get acetyl-CoA coming off of this with a reduction of the lipolysine being formed from this reaction here. FAD picks up these protons, is reduced to FADH, and then the NAD picks up these protons right here. So this is a reaction that occurs in the first reaction of pyruvate going to acetyl-CoA. This is what coenzyme A looks like. It has a beta-mercaptoethylenamine moiety, pantothenic acid here, and then the ADP molecule at this point. And this is acetyl-CoA, which attaches to this. So the reactive thiol group is here, and this is where it attaches here. So this is the moiety that is attached to what had been pyruvate. The intermediates never leave the complex. Among the reactions of citric acid complex, the acetyl-CoA donates the acetyl group to oxaloacetate to form a 6-carbon citrate molecule that is isomerized to isocitrate and decarboxylated to a 5-carbon alpha-ketoglutarate. Then is decarboxylated again to form 4-carbon succinate, then converted to oxaloacetate, which re-enters the cycle. This is a sequence in plain English. We will look at this mechanistically so we can understand how we get from acetyl-CoA and oxaloacetate to oxaloacetate coming out the other end of the cycle with loss of two decarboxylations here and here. The mitochondria also oxidize fatty acids and amino acids to acetyl-CoA, and at one time, evolutionarily speaking, mitochondria was a facultative bacterium which was probably incorporated into the host eukaryotic genome. Complete oxidation of acetyl-CoA is preferable to the alternative of direct decarboxylation, which forms inert methane, and then you would form a carbon moiety instead, carbonyl moiety instead. So it does have its advantages to have acetyl-CoA oxidized. So when we look at the citric acid cycle, we have acetyl-CoA up here with the CoA component right here, and we have what's called a Claisen condensation, where the methyl group of the acetyl-CoA is converted to methylene in citrate, which is right here. This is citrate, and you get hydrolysis of this CoA right here, and this is an energy absorbing step here so you get the citrate molecule attached at this point, and this is stereospecific. After this, you get dehydrogenation and rehydration. The hydroxyl group of the citrate is repositioned in isocitrate, which sets up for decarboxylation, next step. So this moiety right here is transformed as an intermediate of cis-aconitate with rehydration steps. So water leaves, and then it reappears here. This is isocitrate. The next enzyme in the cascade has an oxidative phosphorylation, where the hydroxyl group is oxidized to carbonyl, which in turn facilitates decarboxylation by stabilizing the carbanion formed on the adjacent carbon. So this is isocitrate. We get loss of carbon dioxide, and we get alpha ketoglutarate here. This is a 5-carbon moiety. We lose the carbon from this point here and get oxidation at this end. The next step reintroduces CoA, and this is oxidative decarboxylation. So the pyruvate dehydrogenase-like mechanism depends on the carbonyl and the adjacent carbon. So we have alpha ketoglutarate, CoA attaches at this point, and you get oxidation with the loss of carbon dioxide. The next step takes GDP or ADP. This is a substrate level phosphorylation, where GTP is formed from this, and CoA, the energy dissipated by the loss of CoA, is captured by this step right here. So you get substrate level phosphorylation. The energy of the thioester is conserved in phosphoanhydride bound at GDP. So this energy is a transfer right here, and you get succinate. Then there's a dehydrogenation step with FADH produced, and you get fumarate forming. So this is a trans linkage here. Then you get hydration. This is a addition of water across the double bond, introduces a hydroxyl group for the next oxidation step. So we get hydration of this double bond linkage. And then the next step is a dehydrogenation, where the hydroxyl group is oxidized, and this completes the oxidation sequence, and you generate a carbonyl position to facilitate the Clason condensation in the next step. So this is oxaloacetate. And there are enzymes that mediate these, and we will be covering these now in sequence. So the first reaction is formation of citrate. This is a spontaneous reaction caused by citrate synthase. You have oxaloacetate. You have the aspartate, which forms an adduct with acetyl-CoA right here using histidine linkage. Always keep track of where the electron pairs are going. There, double bond, pick up a hydroxyl group, and the positive charge, the electrons return to this point, stabilized through this aromatic ring here. You have oxaloacetate, and then the reaction reverses, and this double bond that's formed picks up there, and you have another histidine over here that stabilizes it, and you get the enol intermediate, again anchored from aspartate and two histidine molecules right here, and this forms citrate. So in the next step, the enolate rearranges, attacks the carbonyl carbon of oxaloacetate. Histidine is positioned to abstract the proton it had donated previously, and the histidine at the 320 point of the enzyme, 320 going from amino terminal to the carboxy terminal. That's a general acid, and you get acetyl citrilyl-CoA from this. So you have this situation here with citrilyl-CoA, and then this bond right here is hydrated with liberation of CoA. This is a thioester right there. It's hydrolyzed, and you get the formation of citrate from this, again anchored by aspartate and two histidines. And you can see how this reaction proceeds. Simple hydration step. Then the next step is isocitrate formed via cis-aconitase. The enzyme is aconitase, and this is a dehydration step with formation of an alkene linkage here. This is an unstable intermediate. It is rehydrated, so this basically flips the order of the proton and the hydroxyl group, getting isocitrate with the proton at this alpha position, with the hydroxyl rather. Number 3 step is isocitrate oxidized to alpha-ketoglutarate and carbon dioxide. The enzyme is isocitrate dehydrogenase. And in this formulation, you get oxidation of isocitrate by hydride transfer to NADH or NADP, depending on the isocitrate dehydrogenase complex. So you get a carbon dioxide moiety leaving here. Again, this is a manganese right there. This is an oxalosuccinate intermediate step. Carbon dioxide leaves. Decarboxylation is facilitated by electron withdrawal by the adjacent carbonyl and the coordinated manganese right here. And then the reaction goes back, picks up a proton, and now you have alpha-ketoglutarate. This is a rearrangement of the enol intermediate. And then you get alpha-ketoglutarate on this end. Number 4 step is alpha-ketoglutarate dehydrogenase complex. In this case, you're oxidizing alpha-ketoglutarate to succinyl-CoA and CO2, analogous to pyruvate dehydrogenase that we saw earlier. It's a complex reaction, and the enzyme is alpha-ketoglutarate dehydrogenase. This is the structure of alpha-ketoglutarate going in. Again, CoA kicks in and ultimately replaces this with the histidine intermediates. NADH is formed, so there is a reduction step here, oxidation here, and you get succinyl-CoA. This is an energetically favorable reaction, again with liberation of CO2. To show this again in schematic form, this is using the pyruvate again as a model. It's the same model. This is the CO2 liberated as hydroxyethyl TPP with stabilization of cysteine, not histidine, but cysteine reaction. This intermediate then adducts with cysteine, which is formed after this linkage is reduced. You get an adduct here, and CoA replaces this bond right there, and this is with the second component, which is a transacetylase, and then in the dehydrogenase you get FAD and then NADH. The next step is succinyl-CoA converted to succinate. The enzyme is succinyl-CoA synthetase, and GTP is formed and converted to ATP. So again, we start with succinyl-CoA here. Phosphate effectively replaces the CoA at this point with histidine stabilizing this right here. Then succinate is formed. And then the phosphohistidyl enzyme with phosphate attached lends its phosphate and forms GTP from this, and succinyl-CoA synthetase is over here. The next step is succinate oxidized to fumarate, and the enzyme is succinate dehydrogenase. FADH is formed, and we get a simple oxidation of this intermediate with FADH formed, fumarate in translinkage, energetically neutral case. Then the next step is fumarate hydrated to malate. This is catalyzed by fumarate hydratase and is stereospecific for the trans isomer of fumarate. So this is simply hydration of the linkage here. There's a carbanion transition state right here, and then you get proton and you get malate forming. And this is a stereospecific isomer. You can see the projections. Malate is then oxidized to oxaloacetate by L-malate dehydrogenase. NADH is formed. So you get a carbonyl form with a reduction of NAD. These go in oxidative phosphorylation. Oxaloacetate is formed, and the this is nearly 30 kilojoules per mole free energy. C14 labeling is used to confirm this pathway. The energy of oxidation of the intermediates is conserved. You get 3 NADs, 1 FADH, 1 ATP or GTP. Citrate has a symmetrical structure but reacts asymmetrically, implying enzyme attachment. And the preponderance of the energy yield is from the electron transfer molecules NADH and FADH. You get 3 ATPs from NADH, 2 from FADH, because of where they enter in the oxidative phosphorylation pathway. So this is a summation of the, if you include the yields from glucose going with the dissolution of glucose to its combusted elements of carbon dioxide and water, the transfer of glucose, the transfer of phosphate to glucose to form glucose 6-phosphate cost an ATP. Similarly, the production of fructose 1,6-biphosphate with a PFK also costs 1 ATP. The formation of 2-glyceraldehyde 3-phosphate to 2,1,3-bisphosphoglycerate produces 2 NADHs, and you can either get 3 or 5 formed depending on which mechanism you're using to shuttle the NADH from there. That's and then you generate ATP from 1,3-biphosphoglycerate to 3-phosphoglycerate. You generate 2 ATPs from phosphonopyruvate to pyruvate. You generate 2 NADHs from pyruvate to acetyl-CoA, 2 from isocitrate to alpha-ketoglutarate, 2 from alpha-ketoglutarate to succinyl-CoA, and you get 2 ATPs from succinyl-CoA to 2-succinate or 2 GTPs. Succinate to fumarate is 2 FADHs, and the 2 NADHs are formed from malate to oxaloacetate. So when you add and subtract the ATPs, you get a net of 30 to 32 ATPs per glucose. Again, we're referring to, I rounded it off badly, but it's 2.5 ATPs per NADH and 1.5 ATPs per FAD. Not quite 3 and 2, but you can round up for that. And the number for the NADH in the third step with the formation of the bisphosphoglycerate is either 3 or 5, depending on the mechanism that you're using to shuttle the NADH equivalents to the mitochondrial matrix. So about 1/3 of the energy is conserved from total oxidation of glucose. You get about 976 kilojoules per mole out to 2,840 kilojoules per mole available. And this confers survival advantage. Obviously, having a cycle is advantageous in terms of energetic conservation. The intermediates of these oxaloacetate, for example, provide precursors to many biosynthetic processes. So these are under specific rigorous control. So when you look at that sort of formulation, you've got pyruvate. This is a Krebs cycle right here. Oxaloacetate can be converted to phosphoenolpyruvate, which can go up to glucose. From PEP, you can go down and form serine, glycine, cysteine, phenylalanine, tyrosine, tryptophan. You can go in this direction with oxaloacetate and get aspartate, asparagine, pyrimidines. From this, pyruvate can be transformed to malate directly with malic enzyme. Succinyl-CoA can give rise to porphyrins and heme moieties. Alpha-ketoglutarate, having one extra carbon, can be transformed to glutamate, which goes to purines, also to arginine, proline, glutamine, and citrate can be converted directly. Since it's heavily reduced, you can go to fatty acids and sterols quite nicely. There are anapyrotic reactions that replenish these intermediates. So you can have phosphoenolpyruvate convert oxaloacetate, pyruvate to malate, pyruvate to oxaloacetate. The pyruvate conversion oxaloacetate is quite interesting. It requires pyruvate carboxylase, which utilizes biotin. And I will show that in this slide right here. Okay, so this is the reaction that goes shows biotin's use in the reaction, which is characterized by a pyruvate carboxylase. So pyruvate carboxylase is here, and there are 2 catalytic sites, 1 and 2. So in this case, you have bicarbonate, and you have the presence of ATP right here. And the bicarbonate is activated by ATP, and you get a carboxyphosphate, which is shown here. So this oxygen, the electron pair, removes the phosphate terminus, and you get a phosphate form, carboxyphosphate, which is a short-lived intermediate. You get a conversion of this, and you get CO2 developing. And CO2 then is bonded to the nitrogen of this, which is anchored to a lysine right here. So this is the biotin moiety, and it's right over here. This is the biotin moiety. So the carbon dioxide is attached to it right here. You see that in this form here, and this is a carboxybiotin. So this is biotin anchored to a lysine in the pyruvate carboxylase enzyme. This biotin then will transform or transport. Biotin will transport this CO2 from one active site to another active site on the molecule. And at this point, the biotin will be decarboxylated. Pyruvate comes in right here. So here's pyruvate here, and you get decarboxylation of this of the moiety right here. And then the electron pair from this oxygen acts as a sink, reverses course, and then this bond attaches right here to pyruvate, with the electrons being conserved in this right here in this oxygen in the carbonyl group. So this is a pyruvate enolate, and it reacts with the CO2 right here to form oxaloacetate. You have an enolate formed right there, and the CO2 reacts with it and picks up a proton right here, and then it's released from the biotin molecule at this point, and you get oxaloacetate at this end. So you can get some understanding. It's not simply words. They're being in sequential form. This is the electron flow that requires biotin as an anchoring compound. So the biotin vitamin function is inhibited by avidin found in egg whites. If you cook the egg whites, you don't get this problem, but egg whites taken raw will block and eliminate the function of this vitamin. Regulation of the Krebs cycle. Pyruvate dehydrogenase has product inhibition, so increased fatty acids and can do that. Enzyme can also be inactivated by phosphorylases. The exergonic heat-producing reactions, negative delta H, the isocitrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. These are all regulated by substrate availability, product inhibition, and allosteric feedback inhibition, which is chemical modification. These enzymes exist as complexes within the mitochondria, and they ensure effective and efficient transfer of substrate intermediates. And that's it. Thank you very much.